this is a true turning point in research in retinal diseases. I have not seen advances like this coming to fruition, coming to that stage where the foundation can make such a great difference going forward, bringing those preventions, treatments, and cures to individuals.
The work on curing blindness with these various approaches that we're using today, what to me makes it so important is that it's pioneering new types of treatments, say gene therapies and stem cell therapies, that are going to be useful in a lot of other areas of medicine.
The eye is the window to the brain. It's made up of neurons, which is the same that your brain is made of. So as we understand the interaction of the light-sensing cells, the photoreceptors, with the rest of the neural circuits in the eye, we're understanding what's happening in the brain as well. And that's really important. It is leveraging our information for other diseases as well.
It's a beautifully structured system that allows you to understand complex networks of the nervous system that is a lot more simple in comparison to the brain, where in the brain you have thousands of connections that any given neuron might have, whereas in the retina it's a very simple one-to-one-to-one connection between the photoreceptor and its postsynaptic neurons. And so it's just a beautiful system to study that I'm just very intrigued with.
The reason you can't see very well is that a lot of the cells in your retina, the light-sensitive part of your eye, they're gone. And like every other part of your nervous system, your central nervous system, your brain, your spinal cord, when those things are gone, they're not replaced. There's no, it's not like cutting your finger where the skin heals up after that damage and makes new skin cells and new blood cells and all this. You just don't make those new cells in your eye, in the retina part of your eye. So we're trying to find a way to get more of those cells, grow them up in a petri dish, and put them back. And the first thing you say is, well, where are you going to get the cells? And so where you're going to get the cells is from embryonic stem cells. The beauty of embryonic stem cells is that they can make any of the different cell types in your body, but you have to know how to coax them in that direction. And so what we spend a lot of time with day to day is figuring out how to coax them in the right direction, how to make sure we have what are going to be useful cells that will make replacement parts for you, and then how to get them in the right place, and how to know that we got them in the right place, and then how to test whether they're going to do any good for you.
Our company takes a virus called AAV that will infect people but cause no disease. And we basically remove the normal genes from the virus and insert a normal copy of a human gene. We then package that into what's called a vector, a viral vector. And to the body, when you inject that in the appropriate location, such as into the eye, the virus will go and get inside cells, and then the normal gene will be expressed inside the cell and will replace the defective gene, make a normal copy of the protein, and correct the defect.
It's this conjunction of technology has come together so that we know the genetics of many forms of retinal disease. We have the technology now to deliver the genes. We have the animal models for many of these diseases. It can be put together, and now there's interest in actually supporting it to the clinical trial. So we're at a very special time, I think, in the history of medicine, because this is not just retinal diseases we're talking about. We're talking about many, well, it's basically all forms of inherited human disease are potentially amenable to this kind of approach.
There's a lot of excitement about translating research into clinical cures and therapies, but that's a really challenging process, and I think we forget sometimes how difficult that is. And of course, driving all of that is a huge challenge of raising enough money to support the research that's needed.
The most important thing is do no harm. That valley of death, that translation, is what we call bench to bedside. It's translating the bench research, whether it be in a petri dish, in an animal, or whatever, in an assay, into being able to deliver it to a human being.
A phrase that I use a lot with patients that I see is that there's an army of people working on this. I want people to understand that there are hundreds of scientists worldwide working on these diseases, and they do communicate with each other almost continuously, both interpersonally on the telephone, email, but also through the literature. And it isn't a group of people just working in little 4 or 5 person groups, but it's a real society of people that are working together to try to cure these diseases.
We all achieve greater things when we work together, and the Foundation Fighting Blindness identified that and made their funding available in such a way as it encouraged that collaboration and that cross-fertilization, not just between scientists in the same institution, but across the country and across the world.
This kind of work requires many investigators with complementary, diverse sets of expertise. There's no one person who can do all of this because there's so many elements involved. And that's part of what makes this research so costly and why the foundation really needs support from people.
The fact that the foundation, from the beginning of this technology and from the beginning when we introduced it to the eye, they recognized the importance of it, and they have been extremely supportive for this work. Without this funding, we would not have been able to show that we can rescue Leber congenital amaurosis. We would not have been able to show we rescue retinitis pigmentosa model, and we would not have been able to show that we can and able to rescue the Stargardt model, which is the most difficult model.
There's already 50 to 100 people in this country who have benefited from gene therapy. There are going to be hundreds of people in clinical trials. There is not just hope in the indefinite science fiction future. There's hope today because real progress is being made in real living human beings to bring treatments to the clinic.
I joke with my surgeon friends and I'm saying, well, surgery is going to go out the window. As soon as you can correct the disease with a gene and a vector, a recombinant virus, why go in and cut the thing out when you can do it the way it's kind of reverse, if you will, the way the disease initiated itself in the first place.
And our ability to move that forward is extremely important, and it's what drives me day and night. It's why I work the way I do. It's why the foundation works the way it does, because we know we need to do it, and it is an exciting time. We're seeing advances in ways that weren't seen even 5 years ago.
The main driver is people who are going blind and need a treatment, and they are desperate. The time clock is running, and every single day is one day when they're losing more cells in their retina, and there's less chance of resuscitating their vision. And I can't tell you how motivating it is for me and for my colleagues and my lab members to talk to these people and see the challenges that they deal with on a daily basis.
Especially if one sees children as part of their practice, and you sit in the room with a family and you see a child that is either the age of one of your children or reminds you of one of your children when they were that age, and you realize that they have a pretty bad thing that is endangering their vision. This sense of responsibility to them steals over you, and your desire to press through obstacles steals over you. And so the word fight, I think, is very appropriate, because you sort of you're leaning forward, you're looking at this obstacle, and it's sort of like, if you're between me and the vision of this child, you don't want to be between me and the vision of this child.
All of the subjects in our clinical trial have asked whether they can see the lab after they've had their gene therapy. And I've brought them all to the lab and introduced them to the person who cloned the virus and the person who developed the immune testing, people who've run animal studies. And it's been so meaningful to the scientists in the lab to meet these people. And many of them have had their photographs taken there, and we have them hanging in the lab. We get letters, emails, phone calls every day, hundreds of them. And we answer all of those calls. All of these people have their own unique stories. And we have the pictures mounted on the wall, people who've asked us to share their story and look at my beautiful child who really needs help. And these things are just so incredibly motivating.
If I can just be a part of the process that one day gets a patient who is fighting a disease like retinitis pigmentosa to see better or to preserve the vision that they have remaining, then what more could you ask for in your whole career? Very few people, whether they be physicians or scientists, are able to say that they can play a role in something as great as that.
Venture philanthropy, I think, is an accurate description of what the foundation does. As you know, Gordon Gund and Steve Wynn have retinitis pigmentosa. They have been able to leverage their own funds and funds of others to do the research that's going to lead to products that will help people with a variety of retinal diseases.
We have projects lined up just waiting for the resources in order to take them forward into the clinic. And that's really where donors can make a huge difference for us. We have that taxi line just waiting, one after the other, and as we get the resources to move them forward, that's what we do. In a scientifically sound way, in a way that's going to make a difference for those individuals. We just need the resources to be able to do it.
A donation equates being able to buy a necessary piece of equipment that I couldn't purchase otherwise. Would allow me to pay a stipend to a student that would like to join my lab to work on some of these important questions that I couldn't hire otherwise. Allows me to pay my mouse bill. Allows me to buy chemicals, whatever else I need. It keeps the lab running.
If we had lots of people raising those $57 of the cookie sale, we'd have enough money to really fund a research project. And maybe that's not going to fund the next clinical trial, but if you put a lot of those, 1,000 of those together, maybe you'll fund the next laboratory-based project, which FFB I think is really best at funding, that will identify the next potential therapy for another disorder. It's additive. All these efforts come together can really make a difference.
They all add up.
The point I would make to any donor, whether it is a John D. Rockefeller or a $10 donor, is we are making a difference such that a parent, an affected individual, a child, should never have to hear you will go blind. All they will hear is, you have a retinal degeneration, we've got a treatment for that that will make sure you don't go blind.